Now, in terms of fluid shifts between compartments, all right, so we're talking about the intracellular versus the extracellular compartments. All right, so factors that affect fluid movement are one, the membrane permeability. All right, so how permeable is the membrane to to water or ions, and forces acting on the fluids. And by forces, what I'm really talking about is diffusion. All right, so concentration gradients, and really a subtype of diffusion, which is osmosis. Okay, membrane permeability. In terms of membrane permeability, a couple things to keep in mind. First off, generally speaking, cells are freely permeable to water. Now that's generally speaking. There are some very key exceptions to that, and generally the exceptions, most of the exceptions I'm going to talk about in terms of general rules are going to occur in the kidney. Okay, so for the most part, cells are freely permeable to water, effectively impermeable to sodium, potassium, chloride, glucose, and so on. In other words, they're going to need specific transporters, and they can't just move across the membranes easily. But membranes are freely permeable to things like oxygen, CO2, urea, glycerol, basically any kind of small lipid soluble molecules, as we mentioned, or as I've mentioned earlier. These can move across that membrane pretty easily. So we need to understand what's permeable versus what's not so permeable, because that's going to have a great impact on water shift. Okay, so now in terms of the plasma membranes and being selectively permeable, we kind of discussed this a little bit. Okay, but keep in mind they're always permeable to small uncharged molecules or moderate-sized nonpolar molecules. Okay, transmembrane proteins that act as channels or transporters increase the permeability of the membrane to certain compounds or ions. And so you can see down here in the cartoon, if that circle is the cell, you'll notice the concentration of potassium is about 155 millimole compared to outside, which is about 4 millimoles. These are actually normal physiological ranges. So I have a gradient high inside the cell to outside. Sodium 140 to about 5 or 10 millimole inside the cell. Again, we have a gradient wanting to go inside the cell. Here's my chloride concentrations, my glucose. Right? Now you'll notice in the red ones, you'll notice it's kind of bouncing off the membranes there, just to kind of show you that it's effectively impermeable unless those channels are present and open. Urea, H2O, O2, for example, they can pass readily. Okay, for the most part, they'll move through cells without issue. Very basic in terms of gradients, all right, across that plasma membrane. They are due to differences in their permeability. Okay. If sodium were freely permeable, there would be no gradient. But since there is a difference in its permeability, or at least selectively permeable, we can create gradients. So this concentration gradient, right? The difference in concentration of a chemical between one side of the plasma membrane and the other, as I mentioned. And this is just a silly cartoon right here, but it's going to help kind of give you the visualization. That's, at high concentrations, which is our guy up here on top of the hill, has a lot of potential energy. Okay, and then he wants to go down his concentration gradient. Okay, so he's going to go downhill, down his potential energy gradient, if you will. And then there's the up the gradient. The up the gradient would be basically being carried out, or moving molecules against their gradient, carried out by those active transporters, which I've mentioned. This should just show you generally how gradients in physiology occur. And I bring this up because anytime I see patterns, I like to show you the pattern because you're going to see it repeated multiple times throughout the semester. So for example, we can have pressure gradients, high pressure and low pressure. Pressure always goes from, for example, in the movement of fluids through your cardiovascular system, fluid or blood moves from high pressure to low pressure. Okay. High concentration to low concentration, which we've talked about, all right, which you can see there. But heat, right? We go from heat, so high temperatures, to low temperatures. So even the thermal gradient is there. We have high temperature to low temperature. So it always wants to go down that gradient. Just in brief, we kind of discussed this already, but transport across that plasma membrane. So processes that move the substance across the cell membrane again are passive and active. Simple, facilitated. Osmosis, which is really a form of simple diffusion. Again, that's going to be a passive process. And then the active process is going to require ATP, moving things against their gradient, which have primary and secondary characteristics, which we've discussed, as well as things like vesicular transport. So this is where you're moving like bulk or very large molecules into or out of the cell. More detail on that to kind of follow up in the lectures to come. In terms of simple diffusion, so here's an example. Here we have a vial of water, and they put a little tablet at the bottom there, of dye, blue dye there, and you can see that's going to have very high concentration there at the bottom of the vial. As those molecules kind of bump into each other and move around, they're going to start to disperse, and that's when you see this diffusion as it kind of goes through the water column there until it's evenly dispersed, and there's no longer a gradient. So in A, in the beginning, we have a high gradient, high concentration at the bottom of the vial and very low at the top. All right, but then we're at equilibrium ultimately. Okay. Now, this is really a random mix of particles due to their kinetic energies. They're moving and bumping into each other. This diffusion is influenced by several things, right? The steepness of the concentration gradient. So the larger the difference, okay, the greater the energy. Okay. Temperature. In our bodies, that's going to be fairly well maintained. It doesn't fluctuate all that much, but there can be some differences, particularly very high temperatures or fevers. The mass of the diffusing substance. Larger molecules will diffuse a little bit slower. Surface area. So diffusion, say something like oxygen and CO2, that becomes particularly important, say in the lungs. So we want it to be able to diffuse in and out of red blood cells, for example, and into the alveoli of the lungs, and the oxygen to go into the lungs and into the red blood cells. We want them to be able to diffuse readily, and so having a large surface area in the lungs will help to get more oxygen and release more CO2. Diffusion distance. So this is sort of intuitive, but you know, it takes longer for diffusion to take place, the longer it has to travel to get to inside the cell. Another example of this would be in the lungs again. If, say, I had a lot of scar tissue in the lungs, around the alveoli, or fluid in the lungs, that creates more distance that, you know, oxygen or CO2 molecules have to travel to get ultimately to those red blood cells, or get from the red blood cells into the alveoli. I've increased the distance, it increases diffusion time, and this can really impact somebody's well-being and ultimately their ability to oxygenate appropriately. So in terms of simple diffusion, the reason I discussed it is because osmosis is really just simple diffusion for water. Oftentimes when we're talking about diffusion, we're talking about the solutes, okay, how the solutes move, you know, the things that are dissolved in the water. The solvent itself, the water, all right, when it goes down its concentration gradient, all right, we call that osmosis. It's the same principle. Water in high concentration wants to go down its concentration gradient. Anytime water goes down its concentration gradient, we refer to that as osmosis in its simplest terms. So it is inversely related to the solute concentration. So what does that really mean? It just means that if there is a lot of solute, let's say inside the cell relative to outside the cell, there's just not enough space for all the water molecules. So there's less water where there's a lot of solute, because the solute takes up more room. So there's lower amount of water when there's a lot of solute present. And I'm going to give you an example of this in just a minute. So osmosis, I mean, this is the force for the diffusion of water under specific conditions, which I'm going to show you some examples of. All right, so okay, so let me draw out an example for you here of osmosis. All right, so I'm going to draw a box. In that box, I'm going to have side A, side B, separated by a semi-permeable membrane there, which I'm trying to draw as a dotted line. And I'm going to draw some substance dissolved in the water in B, all right, and then some dissolved in A. So now I have a certain number of molecules here, a certain concentration of molecules in A versus B. You can see that B has a higher concentration of whatever that substance is compared to A. Okay. Now the question is, you know, the first thing is, are these molecules able to move across that membrane? And so I'm going to let you guys know right now that this green molecule, whatever it happens to be, cannot cross this barrier here that separates A from B. They are not permeable. And I have a higher concentration or a higher amount of those molecules inside B than I do side A. So the question is, will water move, and if it does, which direction? So if you look here, if you recall that water moves towards the higher solute concentration, then you would know that water is going to move from A to B. Okay, we're going to move from box A to box B because there is a higher solute concentration. So a general thing to think of when you think of osmosis is think that water always moves towards the higher solute concentration. So in this case, since B is higher, water moves towards B. The reality of what's actually happening is water is moving down its concentration gradient. Because there's more solute molecules in B, that means there's less room for water molecules. There are more water molecules on side A, so it's really, in reality, water going down its concentration gradient. But I find it easier to think of it as water just moves towards the higher solute concentration. All right, so in this case, that means water is going to move from side A to side B. So let me draw another box over here. All right, so this is the same box. All right, I'm just moving over so I can draw additional pieces here. So let me draw in those molecules. All right, whatever those happen to be. Now we know as the water was moving in, or it moves into this side, it moves into side B. What happens to the pressure in the container inside B? Let's say those are rigid walls. Okay, as water moves in there, what's going to happen to the pressure in B? Well, the pressure is going to go up. What happens is we increase the pressure that's exerted on the walls of that container as it starts to fill up with more water. Okay, now what's going to happen? Well, as the pressure increases along the walls here, as water is moving in, water will ultimately stop moving in when the pressure reached is high enough to resist water moving down its concentration gradient. So when the pressure is high enough, the pressure exerted against the walls is high enough to resist water moving down its concentration gradient, we reach an equilibrium now. All right, this is actually referred to as osmotic pressure. Osmotic pressure. So to start, in phase one, what you had was water moves in from side A to side B into that container in B there because it's moving towards the higher solute concentration. Part two, though, what ended up happening was the pressure inside B's container there started to increase. This is a pressure often referred to as hydrostatic. Hydrostatic pressure, this pressure that's being exerted against the container walls there. As that pressure increases, as that hydrostatic pressure increases, it starts to resist the osmotic pressure, which is the pressure of the water moving down its concentration gradient. And eventually they'll reach an equilibrium where the hydrostatic pressure, or the force against the walls, is equivalent to the osmotic pressure, or the pressure of water moving down its concentration gradient. At that point, we reach what we call an osmotic equilibrium, or an osmotic pressure. And so we can actually measure that pressure. Now, usually our body is in what we call an osmotic equilibrium, meaning that the pressures inside and outside of the cells would be equivalent. Let me give you another example here. So here, if I draw out, I'm going to draw another square, but I'm drawing it a little bit taller, so the columns are a little bit taller. Okay. I'm going to draw a little bit of a cube like that. In fact, you know what? Let me draw a dotted line here to represent a semi-permeable membrane. I'm again going to have column A and column B. And I'm going to tell you that this represents, this line right here, represents the water level. All right? In other words, up here is air. This is just air-filled space in this part of the container. This level right here is water, and this is water down here. So I have, let's say, just water right here, and then I have dissolved in the water on side B. I have some molecules. I'm going to draw a few in here. All right. Now again, these molecules are not permeable, meaning they cannot cross over into side A. I have nothing dissolved in the side A compartment right now. So the question is, where is the water going to move? Well, it's going to move from A into B towards the higher solute concentration. As it does so, what's going to happen to the column of water on side B? Well, it's going to start to rise. So I'm going to redraw and show you that the column of water is going to rise on side B, and it's going to be lower on side A. Because as the water moves from side A to side B, it loses that water, so that level drops, and the side on side B is going to elevate. As the level of water starts to accumulate on side B, you're creating a larger column of water. That's going to exert a downward pressure, similar to what we talked about in the boxes to begin with, these boxes up here. With the hydrostatic pressure, we're going to have downward pressure, similar to that pressure against the container. Again, it's a hydrostatic pressure. A hydrostatic pressure. Now, that's going to resist water moving in. So in other words, water will only be able to move in just so much because the hydrostatic pressure, or the pressure from the weight of all that water that's accumulating, is going to act to resist any more water moving in. And so therefore we're going to reach an equilibrium. Again, that's going to be that osmotic pressure. And I'm going to draw it one more time because I want to make another point here, is this. Let me draw that semi-permeable membrane, side A, side B. All right, now I'm going to draw my water level back to where it was. And I'm going to draw more molecules on side B there. So I've actually increased the number of molecules on side B. I'm not adding anything to A at this time. So if I were to simply ask again, where's the water going to move? It's going to move towards the higher concentration, which is towards side B. And as you guys know already, that's going to change the water level because B's water level is going to rise as A's is going to drop. Now you'll notice here I drew the water level higher. The reason is this, is that the difference, there's a much higher solute concentration inside B. The number of solute molecules there is much greater than it was in the first example I gave, which means it's a steeper gradient. That means water is going to move much more readily into that space. In other words, yes, water moves towards the higher solute concentration. The higher that solute concentration is, the greater the water is going to move down its gradient into that space. Okay, so in this case, in this example, water much more readily moves in and it increases the column even further. So in fact, the hydrostatic pressure has to elevate even higher than it would in the first example in order to counter the osmosis that's occurring. So the osmotic pressure in this example, the osmotic pressure is greater than the osmotic pressure in this example. It took more hydrostatic pressure to resist water moving across in our example right here than it did in our example over here. So basically, the concentration of impermeant solutes, the higher the concentration of impermeant solutes, means the greater the osmotic pressure. Sometimes it's looked at as sort of the pulling force, if you will, of pulling water into that space. If I have a higher concentration in column B, so if I were to increase it still further, then I would draw even more water into column B. All right. The last thing I want to touch on here, and I'm just going to erase part of this so I have some room to draw. All right. Let's get this all out of here. Is, let's say, as an example, I have my square again. Here's A, and here's side B. let's say I had, greater concentration on side B. However, in this example, the solute molecules are permeable. They're permeable. So in other words, these molecules can actually move across the membrane and go to side A. And in fact, what's going to happen is if I draw that again over here, again, I apologize, I'm just drawing it quickly to make the point. Here's column A, here's column B. Let's say that happens. What's ultimately going to happen is they're going to reach an equilibrium. They're going to go down their concentration gradient and equilibrate with column A because they are permeable to that membrane. They'll do that fairly rapidly, and will I have any fluid shift now? And the answer is no. Fluid will not move anymore because the fluid, yeah, it did move, it equilibrated itself as well, but since the solute also moved with it, all right, the water level didn't change. All right, it didn't change because the solute molecules had actually re-equilibrated themselves across, so the water levels would be equivalent on either side. So it didn't change. Now, so in this case, this is really to point out the fact that what's going to cause fluid shifts are differences in concentration gradients of the impermeable solutes. Permeable solutes will not have any effect on the fluid shift, but the impermeable ones will. So I'll come back to that. So that's basically osmotic pressure, and this example here in the cartoon is going to kind of illustrate that again, where the osmotic pressure is proportional to the concentration of the solute particles. All right, so here you can see in this cartoon you have a U-tube. And so in the U-tube, all right, we have the left arm and the right arm, and it's separated down here by the semi-permeable membrane. And you'll notice on one side of the membrane are various solute molecules. They're impermeable, so they can't cross over that membrane to the other side. And so we just have pure water on the other side. So we have osmosis, right? And we're going to have water moving down its concentration gradient, or in other words, we're going to have water move towards the high solute concentration. And the column on the right arm of water starts to go up, and as it goes up, that's going to exert a hydrostatic pressure that's going to oppose the osmotic pressure. Oh, excuse me, it's going to oppose osmosis. And so what we have is our hydrostatic pressure opposing the osmosis. So you see two equal arrows there to indicate osmosis going towards the right and hydrostatic going towards the left. And we've reached an equilibrium there in terms of pressure. Now we can measure that. So if this were in a lab, we could actually have this little piston here connected to a pressure transducer. Just push it down to see how much force we had to exert in order to push that column back and force water back across the membrane. All right, it doesn't want to go that way, so we have to actually physically push this column down to see if we can force the water back across that membrane. And how much force did that require? That's what we call our osmotic pressure. That's how much force. All right, that's how much force was required. So we can actually see that. What was that applied pressure? All right. So the osmotic pressure again is equal on both sides of the membrane in our body under normal healthy conditions. Okay, on either side it's equal. What we have in normal circumstances in our body is inside the cell we have various solutes, some of which are impermeable, which will affect water shift. But outside the cell we can have different particles. As long as it's equal in number, it'll prevent any fluid shifts or drastic fluid shifts moving inside or outside of the cell. Okay. So here's, now clinically we're going to talk about osmolality and tonicity. What's the difference really? Again, remember I use osmolality and osmolarity interchangeably. So osmolality refers to the moles times the number of particles, which is what I reviewed with you guys, because the higher the number of particles, the more it's going to draw water towards itself. Okay, and we refer to that as osmoles, and it's really dependent on those impermeable solutes. So we use three terms to compare concentrations of one solution to another. Okay, and keep in mind, our reference solution is our intracellular fluid. All right, so we're comparing any fluids that, let's say, we give to the body, we're comparing it to our intracellular fluid osmolarity or osmolality. So for example, we can call it hyperosmotic if the osmolarity or osmolality is higher than ICF. Isoosmotic if it's the same, and hypoosmotic solution if it's lower than the intracellular fluid. So for example, the ICF, a typical osmolality is going to be 285 milliosmoles. 285. All right, so that's a fairly standard. There's a range, of course. Isoosmotic solution, if it's 285, would be the same as the intracellular, so we call that isoosmotic. Hyperosmotic would be anything greater than 285 in this case. Relative to the ICF would be hyperosmotic. Anything lower than 285 would be hypoosmotic. Right? So if I had a solution that was 260 milliosmoles, that's hypoosmotic relative to the inside of my cells, which is 285. Tonicity is a biological term. This indicates what the size of a cell will be after the solution produces any shift of fluid. All right, so I'm going to show you some of experimental concepts with this, but this pertains to actually giving solutions to people as well. Whereas I compare initially the solutions to each other. So I know that, let's say the person's osmolality is, say, 285 milliosmoles. Okay. Now understand that means that if the inside of the cell is 285 milliosmoles, it's 285 milliosmoles in the extracellular fluid as well, because they're always in a balance with each other. Okay, so whether it's inside or outside the cell. Now, if I want to give fluids to a person, and the fluids are 400 milliosmoles, that's hyperosmotic relative to the body cells, right? So that's comparing osmolalities. Tonicity, on the other hand, is what happens after I give that solution. So let's say it was hyperosmotic and I gave that solution to the patient. What happens to the cells in response to that? Okay, so that's really important to understand, and that's what I'm gonna give you some examples of that. But what happens to the cells after it's introduced to that solution? So what size will they be after the fluid shift? So in other words, if it's a hypertonic solution, this causes a cell to lose water and to shrink. Meaning the solution I gave has a high concentration of solutes that drew water out of my cells. All right, and it causes that loss of water to make the cells shrink. That's what we call crenation. Okay, isotonic means there was no change in water shift at all. Okay, and hypotonic causes the cell to gain water and swell, meaning that water was drawn into the cell and it blew up like a balloon. So let's talk about some examples. Okay. So let's try some examples of fluid shifts. I'm going to use an example that is actually in the notes that I have provided for you for the lecture. So I'm going to draw a beaker here, and inside that red beaker I'm going to have a single red blood cell. Obviously this is not to scale, red blood cells are very, very small. But here we have the red blood cell, and inside the red blood cell the osmolality would be 285 milliosmoles. Okay, 285 milliosmoles would be what you'd find in any normal cell throughout the body, including the extracellular space, since everything is in equilibrium under normal healthy conditions. Now, in the red blood cell, we have high concentration of potassium, we have anions, which I'll denote as an A- there, things like phosphates and so on, proteins, things that can't get through the membrane readily, so they're impermeable. So 285 milliosmoles is really the effective osmolality. All right, these are the solutes that are really cause or have an impact on water movement. So in this example, what I want to do is I want to add a solution to that beaker. Let's say the solution that I'm going to add is 285 milliosmoles as well. It's going to be 285 milliosmoles of sodium chloride. Okay. Now, the first question I need to ask is, what is the osmolality of that solution that I want to add? And to answer that question, I need to compare it to the intracellular osmolality of the red blood cell, because that's our reference, is what's inside the cell. So our reference is 285. So if the solution I'm going to add is also 285, that makes it a isosmotic solution, since they're equal. Now the next thing I'm going to do is I'm going to add that solution to the beaker. When I add that solution to the beaker, that's my 285 milliosmoles of sodium chloride. Okay. The next question is then, what is the solute inside that solution that I've added? And really what I'm asking is that solute permeable or impermeable to the cell membrane? Sodium chloride is effectively impermeable, meaning that it's not going to get through that membrane very readily. Okay, so in that case, I have an impermeable solute. Now, so the next question I have now, my third question, is what is the tonicity? The tonicity, if you recall, is really asking what happens to the red blood cell in the presence of that solution. Is it going to get larger? Is it going to shrink? And that all has to do with whether or not water is moving. So in this case, when I've added an isosmotic solution of sodium chloride, which is an impermeant solute, is there any water shift? And the answer is no. In fact, I have equal, both 285 inside and outside of the cell, of impermeant solutes. And so there's no net movement of water in this situation. And so since there's nothing that's going to occur to the red blood cell, we refer to that as isotonic. And in fact, when we give solutions to, say, a dehydrated patient in the ER, right, we put that into their IV, we'll usually give something called 0.9% normal saline, which I've actually discussed on previous slides. And 0.9% normal saline, if you do the calculations, actually comes out to 285 milliosmoles, which matches the osmolality of our red blood cells. And so there's no fluid shift. It doesn't affect the red blood cell, which is what we want. Let's try another example. Okay. Now this time, I'm going to add, let's say, 400 milliosmoles of sodium chloride. So remember the first question when I'm comparing the solution, all right, how is this solution's osmolality compared to the cells? Well, you can see that it's higher, so it is hyperosmotic. All right, so this is a hyperosmotic solution. The next question is, what is the solute? Well, it's again the sodium chloride, which is an impermeable solute. I'm going to add that now to the beaker. And the question is now, what's the tonicity? The tonicity, again, means, is there a fluid shift? Sodium chloride can't permeate, so we have a higher concentration of solute inside the, or I should say in the beaker as compared to inside the red blood cell. And remember, water moves towards the higher solute concentration. Or, in other words, it moves down its concentration gradient. So in this case, water would leave the red blood cell towards the higher concentration. What ends up happening effectively is that as the water leaves the red blood cell, the concentration within the red blood cell will rise. It'll go from 285 to something higher, say 300, 310, 350, and so on, as water is leaving it, because it's going to concentrate that solute that's in the red blood cell. And the water being added to the beaker is going to slowly dilute the concentration that's there until we reach an equilibrium. And in this case, without calculating, let's just say the equilibrium comes out to, say, somewhere in the middle, like right around 350 milliosmoles, let's say. Okay, it would obviously be much less than that. Wouldn't actually affect the 400 milliosmoles all that much, because red blood cells are very small and don't contain a whole lot of water, not enough to shift it. All right, but that would be the general trend. As water leaves, we concentrate the inside of the red blood cell, and as it goes into the beaker, it's going to dilute what's there until we reach a new equilibrium. Okay, obviously this is an experimental condition. We're not taking into consideration the actual body, which would deal with some of this through the kidney. So we're just ignoring that factor for now. Okay, let's try another example. Oh, and by the way, since I didn't mention it before, since it's causing water to leave the red blood cell the tonicity means, in this case would have been hypertonic. So the solution is hypertonic to the red blood cells, meaning it's going to draw water out and the red blood cell will then shrink. As it shrinks, the process of shrinking, we call that crenation. All right, and this is the term for that. So let me write that for you. Crenation. That's when the red blood cell is shrinking. Okay, so let's go with another example. So let's say this time the solution is 600 milliosmoles of urea. All right, so now we're just comparing the osmolalities of each. And you'll notice that the 600 milliosmoles is definitely greater than 285 milliosmoles inside the red blood cell. So this would be a hyperosmotic solution. We're going to add that solution to the beaker. Next question is, what is the solute that's being added with it? Is urea. The property of urea is such that it's small and lipid soluble. So it will pass through the red blood cell plasma membrane very easily. So when I add the solution, one of the first things that's going to happen is the urea being in high concentration in the beaker and being essentially zero inside the red blood cell, it's going to move from high concentration to low concentration, which would be into the red blood cell. So it's going to equilibrate. It's going to go down its concentration gradient into the red blood cell almost immediately until we reach an equilibrium. I'm just going to write 600 milliosmoles of urea inside the red blood cell, but keep in mind, let me redraw that arrow. It's going to move down its concentration into the red blood cell until we reach an equilibrium. I'm saying 600 because it's not going to take a whole lot of urea for it to actually equilibrate in that red blood cell. But the reality is, the urea being lost from the beaker would bring down the milliosmoles a little bit. So say instead of 600 milliosmoles, it might be 559 milliosmoles. Okay, and then what's in the red blood cell would be about 559 milliosmoles. It would reach an equilibrium as it goes down its concentration gradient. And again, that's the property of urea being permeable to the cell membrane. All right, this is why we asked this question. So now the question is, what is the tonicity? In other words, is there a fluid shift? Well, let's take a look at our total milliosmoles now. I have 600 in the beaker from the solution that I've just added, approximately. That equilibrated very rapidly with the red blood cell, so there's 600 that's accounted for in the red blood cell. However, the red blood cell still has an additional, say, approximately 285 milliosmoles. So the higher concentration in this case is in the red blood cell. Water is going to move into the red blood cell. So the tonicity of this solution is hypotonic. Hypotonic. The reason it's hypotonic is water is moving into or towards the higher concentration, which would be inside the red blood cell in this case, and it's going to cause the red blood cell to swell. And if it keeps moving in, all right, in fact, the water will keep moving in until it reaches a new equilibrium, or it causes the red blood cell to lyse or to explode or to burst. All right, so that's what a hypotonic solution would do. Now, the reason for that, again, even though this was a hyperosmotic solution, the reason it was hypotonic was because the solute urea is permeable and it equilibrated across the red blood cell membrane, and so effectively the 600 in the beaker and the 600 in the red blood cell canceled each other out, and I'm still left with 285 milliosmoles more in the red blood cell. So the concentration in the red blood cell is greater than what's in the beaker. So the point we want to make here is that it's really the impermeable solutes that will dictate this, will dictate the tonicity. So let me give you one more example of that, and then we'll move on. Okay. So I'm going to say we have a solution of 600 milliosmoles of urea again. But I'm also going to add to the solution 285 milliosmoles of sodium chloride. So we actually have a grand total in this case of 885 milliosmoles in total. So if I'm just comparing total osmolalities of the solution compared to the inside the red blood cell, we have a hyperosmotic solution. As I add that now, remember it's 600 of urea and it's going to be 285 of sodium chloride. Now the next question is, look at the solutes. What are they? So we have urea, which is permeable. So the first thing that's going to happen there is as soon as I add that solution, urea is going to equilibrate across the red blood cell membrane, and I'm going to have approximately 600 milliosmoles of urea inside that red blood cell. Okay, and then I have 285 of sodium chloride, and sodium chloride is impermeable, so it's not going to be able to go across that membrane. What is the tonicity in this case? Well, now you can see that the totals are exactly the same. We have 285 from the sodium chloride outside in the beaker there. We have 285 inside the red blood cell. We have the 600 from urea, which equilibrated very rapidly into the red blood cell, so those would be equal to each other as well. So this is an isotonic solution. Isotonic solution. All right, there is no net movement of water. Despite the fact that it was hyperosmotic, it was still only isotonic. So really the take home from this is that what dictates tonicity is based on the impermeable solutes. In this case, that was sodium chloride, and sodium chloride being the impermeable solute, in this case, matched the osmolality of inside the red blood cell. There was no net shift. We can essentially ignore the the permeable solutes, all right, except in the case where if it's only permeable solutes, then it acts essentially like water, like free water. If I had only free water, water would move towards the higher concentration in the red blood cell and cause it to swell up and to burst. Okay, so this slide is actually just going to summarize what I've just drawn out for you. But ultimately what it comes down to is if the solution contains only permeant solutes, then it's always going to be hypotonic regardless of whatever the osmolality is. So it could be hyperosmotic, hypo, or isoosmotic. It'll always be hypotonic though. It'll always cause fluid to move into the cells. On the other hand, if you have impermeable solutes only, then the osmolality will dictate the tonicity. So for example, if you have impermeant solutes like sodium chloride, for example, if it's hyperosmotic, okay, it'll be hypertonic. If it's isoosmotic, it'll be isotonic, and if it's hypoosmotic, it'll be hypotonic. However, if I have a mix of impermeable and permeable solutes, it's the impermeable ones that are actually going to dictate the tonicity. Okay, so in our bodies, of course, we have a mixture. So the ones that are going to really dictate that permeability will be the impermeant ones. And in our extracellular fluids, all right, those are primarily sodium chloride and glucose, which is why I show you the calculation there at the bottom of the green box. That calculation there, what it does is it represents the primary effective osmoles. I call it an effective osmoles because those are the impermeable solutes that cause water shifts. Okay, so sodium chloride. So in this calculation, I would multiply sodium by 2. Well, the reason I multiply sodium by 2 is that if it's to account for the chloride that would also be present with the sodium. Not always in equal molar amounts, but pretty close. So it's an estimation. So what I do is I draw the plasma, take a look at the concentration of sodium in millimoles, okay, and then I multiply that by 2, and I add it to the glucose concentration in the plasma, which would be, let's say under normal conditions, 90 milligrams per deciliter, and I divide that by 18, and that should come out to about 285, so the exact calculation, which is approximately that's the normal milliosmol in the extracellular fluid, and it would be matched to the intracellular fluid because they're in equilibrium. So it's a good estimation of what the osmolality is in the body. And so you can run that calculation, and what we can talk about in some of the lectures to come is how we can use that calculated osmolality and we can compare it to a measured osmolality. A measured osmolality would be far more accurate. When we compare the two, the reason we might actually do that is simply to see if what we calculate, which should be pretty close to accurate based on the effective osmoles of the body, but if it's, let's say, a lot higher, say it's like 315 is what's actually been measured, and we calculate it's supposed to be about 285. Well, what's causing that additional osmolality? So we're going to start to look for other causes to increase the osmolality. Oftentimes things like alcohols and stuff can increase the osmolality, so it helps us with our differential diagnoses. Okay, so here's where I just want to kind of introduce you to a couple kind of simplified cases just to point out the importance of fluids and fluid shifts. So in the first clinical case, we're just talking about Abby, an infant, who's had bouts of diarrhea, and so the mother had been treating that by giving boiled milk. And of course, anytime you boil a solution, you're evaporating the water, so you're giving something that's very hypertonic, okay, or hyperosmotic, okay, so something very hyperosmotic. Now this is a baby who's already dehydrated, okay, so you're giving a baby who's dehydrated from diarrhea, you're giving them a hypertonic or hyperosmotic solution, and the baby is becoming lethargic and having an altered mental state. And so if you think about it, if I give a hyperosmotic solution, okay, that can actually draw, it can be a very high concentration, that can draw fluid out of your cells. And that can actually further worsen the dehydration that exists there, which is why if it's drawing it out of, say, neurons and things like that, you can worsen their state, and it can actually lead to comas and seizures and so on. In the second case, however, I have an adult, and this is, you can see in the painting over to the right there, this is Samuel Taylor Coleridge's Rime of the Ancient Mariner, right? So this mariner was stranded out at sea, all right, and he has the famous line of, water, water everywhere, but not a drop to drink, right? Sort of the irony of the situation. And then he said, water, water everywhere, and how the boards did shrink, the boards of his ship. So they were shrinking. It was like effectively crenating, if you will, right? Like the red blood cells that I discussed. So he can't drink it even though he's severely dehydrated. And the reason, again, is it's a similar situation. Is you have seawater, which is a very hyperosmotic and hypertonic solution. It's a very high concentration of sodium chloride, and by drinking that, it'll draw water out of the cells and actually exacerbate the dehydration. It can be fatal. So these are two similar cases. What I'd like to do in the next slide is actually go over a way to kind of visually represent the fluid shifts and how they compensate both intracellularly and extracellularly. Okay, so what I want to show you here now is something referred to as a block diagram. A block diagram. Okay, so a block diagram is a good visual on how water shifts occur in the body, all right, and the compensations that occur with them. So what I'm going to draw for you first is just a large rectangle. That's our box, okay? And I'm going to draw slightly off the midline there. I'm going to draw a dotted line. So not quite on the midline. So in the larger box to the right, I'm going to write ICF for intracellular fluid. And over here, this will represent the volume of the extracellular fluid. And the dotted line just represents the semi-permeable membrane that separates the cell membranes from the extracellular fluid space. Recall that the extracellular fluid represents both interstitial fluid as well as intravascular fluid. And of course, the ICF just represents the collective total intracellular fluid in the body. Now, under normal conditions, both the inside of the cells as well as the extracellular compartment has the same osmolality, approximately 285 milliosmoles per liter. All right. Now down here, I'm going to draw a little zero there, and the total volume. And we're going to make this, by the way, this example is going to be of a 70 kilogram person. right? And if you recall from previous calculations that we did in this lecture, the total body water for that is going to be 42 liters. Remember, that's 60% of the body water. All right, in that case, that would be the male's percentage. Okay, so that 42, if you recall, 2/3 of that would be the total intracellular volume. So I'm going to write 28 out here. So down here along this axis represents volume. As we go in the extracellular, the last third would be about 14 liters. So the numbers that I got for this were actually derived from our previous calculations, okay? So you can redo that for practice if you need to. The y-axis over here, moving in this direction, represents concentration. All right, concentration is in milliosmoles. So the top point right here would be 285. All right, so that represents 285 milliosmoles. Okay. So in summary, this box right here depicts the extracellular versus the intracellular volumes, as well as their concentration. And you'll notice that the concentration is exactly the same in milliosmoles. I'm just going to get rid of this for now. The 285 is exactly 285 both in the ICF and ECF under standard equilibrium in our bodies. Now let's talk about what happens when we introduce certain variables. So let's start with the variable of, say, adding normal saline. So I'm going to attach an IV to the patient, and I'm going to give 0.9% normal saline. Well, remember the normal saline, we talked about that. That actually is about 285 milliosmoles of sodium chloride. So the question then is, how does the box change when I introduce this IV fluid? So let me just erase this arrow here. I'm going to move some things over here just to make it a little bit easier to work with. Okay. Remember that that axis right there represents concentration, which I'll just denote as brackets over here. Okay. In milliosmoles. So let's say I have this patient and I give them 1 liter, we'll say, of normal saline. Okay, if I give 1 liter of normal saline, remember I'm introducing that via an IV, so it's going into the intravascular space. And if you recall, anytime we introduce fluids to a patient through IV, it's always going to be in the intravascular space, so the ECF will be affected first. So how is the ECF volume going to change? Well, it's going to go out. So remember, volume is my axis down here. So the ECF is going to expand from 14. I'm only adding a liter, so I'm just going to approximate here. I'm going to draw it out about here. I'm going to draw a box that looks like that. So it's expanded outward to here, which since I'm only adding 1 liter, that would bring it to about 15 liters now instead. Okay, so I went from 14 to 15 because I've just added another liter to the ECF. And the question is, well, how did I know to go to the concentration of 285? Well, I know that because I've added normal saline, which its concentration is 285, which matches the concentration of the ECF as we discussed. So that's what that would look like. If I added 2 liters, I would make the box outward a little bit bigger to 16, 3 liters, and so on. Okay. And the question next would be, would there be a fluid shift at all? And the answer to that, as you guys already know from the previous slides, is that there is no fluid shift, because what I'm really asking is, what's the tonicity, right? How does it affect the intracellular fluid? There wouldn't be any shift in the intracellular fluid because I've added an impermeable solute to the ECF that matches what's already the concentration inside the cell. So there's no fluid shift. So that would be effectively what happens when you add, say, an IV fluid. Okay, so let's try another example. Let's try the example of fluid loss. So I'm going to get rid of what we've drawn here so far. All right, and let me make some room. Okay, so in this next example, let's say the patient is dehydrated from sweating. Maybe they were running a marathon or exerting themselves heavily. And so we have dehydration from sweat. So a key thing to understand about sweat is something I mentioned in an earlier slide, is that we primarily lose more water than solute. So the solute will concentrate. Okay? So let's take a look at what that would look like in our box right here. So first off, if I'm losing it via sweat, any kind of volume loss that occurs, say during sweating or something of that nature, would occur via the extracellular route. All right, so you're going to lose volume from the extracellular space. In fact, that makes an important point right now, is that any volume addition or volume loss first affects the extracellular fluid, and then the intracellular fluid responds. All right, responds to what happens to the ECF. So all changes occur in the ECF first. All right, so with sweat, I'm losing primarily just water with sweat. So let's see what that looks like. If I'm losing water because I'm sweating, that means my volume is going to go down in the ECF. So I'm just going to pick a spot. Let's just say I lost a couple liters here. All right, so let's say at this point I've lost a few liters. All right. So I'm going to draw a line to here. Now the question now is, before I go any further, if I'm losing just water, what happens to the concentration in the ECF? Well, if I lose more solvent relative to the solute, it concentrates even further. So I'm going to have a higher concentration. So I'm going to draw it a little bit above. Remember, this axis represents concentration here. So this is going to be higher than 285. And I'm going to draw it back to that box right there. All right. So this represents right here, this new shaded box represents that I've decreased my volume here and I've increased my concentration here. Because since I've lost essentially just water relative to solute, the solutes that are left behind are more concentrated. So that's what it looks like initially, what's happening to the ECF. Now I told you that the ICF would have to respond to what's happening to the ECF. So now the question is, what's going to happen to the intracellular fluid in response to that? So is there going to be a water shift? And so let me use a different color here. Remember that right now in the intracellular fluid is still 285 milliosmoles. Okay, so I'll just draw that here just to remind you, still 285 milliosmoles. This out here is greater than 285 milliosmoles because it's lost water from sweat. So it has a higher concentration. So water is going to move from inside the cell into the extracellular fluid compartment. And in fact, as water leaves the ICF, the concentration, all right, the milliosmoles here, are going to increase as it loses its water. Those solutes will start to become more concentrated, and it's going to dilute the concentration out here and help to bring that down. And we're going to reach a new equilibrium. It's not going to be our normal equilibrium. It's not going to be 285 anymore because the inside of the cell is concentrating, and the outside we're going to dilute it to bring it down a little bit, but it's not going to go down to normal. And so if I draw what that compensation looks like, I'm losing volume from the ICF, so I'm going to draw it closer to 0. It's concentrating, so I'm going to go above the 285 line here. Okay, and I'm going to draw it across, all the way across here, so it should be straighter than that, but it's going to go further out now and down. So the green box now represents my new equilibrium. We haven't calculated exactly what that equilibrium is in this case, but I'm just representing it so we can think about it logically. So let me review what just happened. Again, the ICF loses water because the water is going to move towards the higher concentration in the ECF, which was dehydrated. Since it's losing volume, that's why I drew the arrow moving in towards 0 there. We're going from 28 to something less than 28 liters. As it loses water, it's going to concentrate, which is why I drew the concentration now above 285 there. And that's going to be at the same level all the way across to the ECF. And you'll notice that's lower than the initial red box that I drew because it's diluting that concentration there. And the volume now has shifted up slightly because now it's gaining water. since the ECF is now gaining some water, it's going to help to dilute that and bring that concentration back down. So although it's still above normal, you notice it's not as high as the red box there, so it's not as concentrated. And now we've reached a new equilibrium. We can call it or make up a number there and just say, let's say it reached an equilibrium around 300 milliosmoles or something like that. All right, so that would be our new working equilibrium there. And of course, this isn't taking into account drinking anything or urinating or the kidneys function at all. Okay, so that's theoretically what would happen. What you guys can try to do at this time is take the clinical cases on the previous slide, all right, and see if you can draw out what in the case of the infant, what the baby had in diarrhea and was treated with boiled milk. What would that box look like? What would be the steps for that? It would be good practice for this. In fact, just so you guys understand, diarrhea, unlike sweating is mostly loss of just water rather than solute. In diarrhea, the loss is usually isotonic loss or isoosmotic loss. You're losing equivalent solute and water amounts. Okay, so the ratio doesn't change at all. All right, one last example, and then I will move on from this. Okay, for this last example, let me just draw out the box one more time. There's my semi-permeable membrane. Here's the ICF. Here is the extracellular fluid. 0, this is 28, 14. Again, this is referring to our 70 kilogram male. Okay. And this will be the concentration on the Y, which is 285, which is equivalent to both the ECF and ICF under equilibrium conditions. Let's say I have something called dilutional hyponatremia. In other words, this is where, let's say, somebody drank just pure water, for instance. Okay. So somebody who had a really high intake of water. So just say, really was drinking a whole lot of water here. So again, it's going to affect the ECF, and that's going to increase the volume, right? So the volume is going to move out in this direction here because they're drinking the water. It goes into the ECF first. And what's going to happen to the concentration? Well, you're drinking pure water, so it's going to dilute the solutes that are there since it doesn't have any solutes of its own. All right, so it's going to bring the solutes down. Now I'm just drawing, I'm not picking any specific numbers here because we're not doing specific calculations. All right, so that's going to bring down the concentration of the ECF, but it's going to increase its volume because I'm drinking pure water, which would be hypoosmotic relative to my body osmolality. How is the ICF going to respond to that? Remember, the concentration in the ICF is actually greater now than the concentration in the ECF, so the fluid is going to shift into the ICF. So when it shifts into the ICF towards the higher concentration, it's also going to cause dilution of the ICF, but it's going to help to increase the concentration of the ECF until a new equilibrium is reached. So let me redraw the line there. So in the end, what will happen is this might come up a little bit because it concentrates a little bit more, but it's diluting the ICF, so its concentration has gone down. Meanwhile, the ICF's volume has gone up, which is why I'm drawing the box going out a little bit further. Meanwhile, the volume in the ECF, I should actually redraw this, hold on one second. So the volume in the ECF will have moved down a little bit. So that new box, the red box, is what that would look like. So let me just describe that again. When you drank pure water, you dilute the ECF, but you've increased the volume. So that's why we initially expanded the volume outwards further out this way. In response, the intracellular fluid, the tonicity of it was that since it was hypotonic, that caused fluid to move inside the cell, which would cause them, of course, to swell. So that's indicated by the green arrow. So water would have moved inside the ECF towards the higher concentration of solute. What that then does is it lowers the concentration inside the ICF, therefore diluting the solutes that are inside that cell, but also increasing its volume, which is why you see the volume expanding further than 28 liters here. since some of that water is leaving the ECF, that would bring the volume down a little bit in the ECF, but it would still be greater than normal. Again, this would all depend on how much the person was drinking, of course. But we're assuming this was a lot of water. All right, so this is what that would look like, and hopefully you guys can see here how this box diagram can actually be a very useful way of displaying what happens in the initial insult in the ECF, how the inside of the cell responds to that water movement, and what the new equilibrium would then be. Understand that under normal physiologic conditions, though, the kidney and our thirst mechanisms would also kick in to help us readjust all this. Okay, the kidney would do so by regulating the impermeable solutes, like, for instance, sodium and chloride. It can also regulate water, and it can regulate the two of them separately, which makes kidneys function quite useful and quite powerful in the body. And we'll get to that later on in the semester. Okay, so this is to hopefully put what we just went through into a little bit of a clinical context. As I mentioned on the previous slides with those box diagrams, anytime you add fluid, right, or you give a patient fluid, it's going to go into the ECF first, right, because you're giving it into the intravascular space, which is part of the ECF. And so if you add the fluid there, it's going to pump up the ECF, and any kind of volume overload in the extracellular space, could increase things like blood pressure. Right? If their blood pressure is decreasing, we give fluids to increase that intravascular volume and bring up that blood pressure. Too much volume in the ECF, of course, can cause volume overload and cause failure of the heart, for example. Now, if there's fluid loss, the loss usually occurs in the ECF first. Okay, so any gain or loss will occur in the ECF initially. So that if there's a loss, it occurs in the ECF first, and then the intracellular fluid will respond to that. But the ECF volume loss, if it's significant enough, can cause a decrease in the blood pressure and can even cause circulatory collapse. So the body is designed to really regulate the osmolality very strongly, and it works in tandem with, they say, the hypothalamus as well as the kidney to make sure that it's regulated very tightly, because large shifts in osmolality can disrupt organ function, like the nervous system, for example, and lead to things like, lethargy, coma, or even death. Okay. In a healthy adult, it's actually quite difficult to alter the osmolality of the body, since it's tightly regulated by both thirst mechanisms and renal excretion. So under normal healthy conditions, it would be very difficult to do. So in fact, an example of this would be if you were to eat a very salty meal, like say a bag of potato chips or something, you're increasing your salt content. That triggers your thirst mechanism to drink more water to dilute that, so it's not increasing the osmolality too much. And so you would dilute that by drinking, and then it's the kidney's job to ultimately get rid of the excess sodium and the excess water. Okay, so that would work in balance with your diet. However, in some cases, some disease states, which you'll learn more about in pathophysiology, you could have a state of, say, hyponatremia, meaning sodium that's too low, or hypernatremia. So in the case of hyponatremia, that would be a hypoosmotic state, since sodium is one of our primary effective osmoles. So it would be hypoosmotic and hypotonic state. That means water would probably be moving in towards the cells and so on. This can happen for several reasons. It could be because somebody's taking in dilute solutions and it's diluting the concentration of sodium that's there. So drinking a lot of, let's say, pure water, for example. Or it could happen because, let's say, the kidney is not functioning properly. It might be getting rid of excess sodium. So if it gets rid of excess sodium, your sodium levels could drop that way too, and it would look also very similar. Now, in terms of hypernatremia, now we're talking about a hyperosmotic and a hypertonic state where water will be sort of driving or coming out of the cells into the extracellular space there, and so the hypernatremia can actually occur because you're adding, say, a hypertonic solution to the body, right? Like in the case of the mariner drinking seawater. Or it could be the loss of, say, pure water, like through sweating, the example I gave with that, which is losing more water than solute. So in those cases, the sodium levels could go up. And then finally, in some cases, you can have an increase in osmolality due to other factors besides sodium. Even though sodium is one of the primary effective osmoles, it may not be the one that's causing the increase in the osmolality. So, for example, you could have alcohols or glucose in excess. So, if somebody is very severe diabetic, has very high glucose concentrations, glucose also being one of the effective osmoles, can cause an increase in the osmolality, even though the sodium level might be totally normal. All right, so these are things that we'll discuss more as we go along, and that you'll definitely learn more about in pathophysiology.